Hey fellas, this is going to be our completion for this slide here. We're just testing this out. Yeah, even here I still get email interruptions. That's awesome. So if you remember, we left off at allele frequency, and allele frequency is how often an allele occurs in a population. So if we're looking, let me use the magic pen. If we're looking here, for instance, 48% of the population is heterozygous, 16% is homozygous for black, and 36% is homozygous for brown, right? But what we have to be concerned of is how many of those alleles occur in the whole population. So let's just take 100 total individuals, right? It means 48 are going to be heterozygous for black. And which one of these is dominant? Think about that for a second. If black and brown are within one individual and it's showing black, then what? Black is dominant, right? So big B is going to be for black. And then over here, 36% of them are going to be, or 36 individuals out of 100 are going to be little b and little b. And we have 16 individuals that are big B, big B. Now what you have to remember is 48 individuals have 48 big B's and 48 little b's. So big B's right now are going to equal 48. Little b's are also going to equal 40. Yay. Go away. 48. If we come in here, little b, that's 36 twice, so that's 72. And then big B is 32 more. I'm going to erase that. You can see this is going to be tough, but we're going to get through it. I'll get better. So 48 plus 32. Now total, if we have 100 individuals, how many total alleles should we have? $200, right? So what's 48 plus 32? It's 70 plus 10, that equals 80 for big B. And then down here, it's 110 plus 10. That's going to equal 120. And collectively, that's 200 total alleles, right? 80 of them, starting around, I can't use my finger, you can't see my finger. 80 of them are, we don't want magic pen. Let's do creative pen. Y'all know this one. 80 of them are big B, and 120 are little b, right? So 80 out of 200, which is what percentage? It's 40%, right? So 40% of the alleles are big B, and the other 60% are little b. So we track that to see if those change, and that gives us population allele frequency. If allele frequency swings from 40% to 45, then we know big B is gaining ground there, okay? All right. Moving on over to this one. So some of you guys thought that mutation was a big deal in terms of evolution, and I'm not saying it's not. But what I am saying is that mutation is not the only source of genetic variation. We talk about natural selection, right? So when organisms are reproducing, they have meiosis, and that creates new offspring in every combination, right? Mutation can add to that, right? So the change in a sequence, mistakes, that's what mutation is, doesn't necessarily affect phenotype, but if it does, it can or cannot have effect on fitness, which, remember, fitness was how successful an organism is at reproducing, right? Then if this mutation makes them more successful reproducing, then it's beneficial, okay? And then in gene shuffling, this is obviously what we talk about in meiosis, right? Sexual reproduction. So those are two obvious sources of genetic variation. How does evolution occur? Well, how do we track these traits, right? So a single gene trait is one where one gene controls the outcome of the phenotype, right? A single gene. This is Mendelian genetics. So it was going to be tall or short. One gene, if it said tall on it, that's it. If it said short, that's it. So widow's peak is an example of that. And in this example, you end up getting a bar graph with either the trait or not. The polygenic traits do something entirely different. More genes, so you get, poly, the term poly's in there, okay? And in polygenic traits, there's multiple outcomes or variations, meaning it's not an either or like single gene traits are. Polygenic traits result in a range of phenotypes where most of the individuals fall in the middle 25%, but we still have these extremes that represent essentially in this bell curve another 25% each for a total of 100, okay? So if they can have possibilities of multiple different alleles, so where Mendel just had tall and short, this is like the rabbits where there were 4 alleles for like chinchilla, Himalayan, the brown fur, or albino. There could be 12, 15, 20, or portions of those where there's hybridization, okay? So in the human example of height, if we use just feet, most individuals fall between 4 and 6 feet, most human individuals. There are individuals that fall under that or beyond that. And there's also blending. So now if we use feet and inches, there's only 12 inches in a foot. So 6'1", 6'2", but then we could break it down to parts of an inch. And if we use the small enough part, nobody is exactly the same height as anybody else if we use small enough portions of an inch, okay? So I think that's enough for our first presentation. So what I want to see you guys do is to answer this assessment 17-1 and do that like you traditionally would do. And in the end, though, this is the change. You're going to have to take a screenshot of your homework, and you're going to upload that using the e-locker and the directions that I sent you in the email. So just screenshot this and whatever type file that is. I hope it's a JPEG or something that's easily read. If not, just convert it to a PDF and then post that in that e-locker, and I'll check your homework that way. All right? Let's be flexible, pliable. I'm going to get better at this. You're going to get better at learning, and hopefully if we do our job, we'll have an opportunity to be back in this classroom where we feel a lot more comfortable and push the curriculum a little bit further. All right, so chins up, make this happen, and then we'll start talking about tests and all that as we go, okay? Just look for email for instructions. This is just a little attempt at making my video here, all right? Talk to y'all soon. Later.